I'd like to thank Susan for the kind introduction and the organisers for inviting me to speak at this symposium. I've also been funded by Basser for some of the work I'm showing here. I'm very grateful for that very important support for this work, which has made a huge difference. So our story begins a long time ago when I found myself staring at some peculiar patterns of BRCA1 down the microscope. It forms very characteristic nuclear foci in S phase. And we can see here in cells marked both with BRCA1 in green and with PCNA, the processivity factor for replication machines, in red, that these foci do not co-localize. But to my surprise, I found that if you stall replication genome-wide by treating cells with hydroxyurea, which depletes the nucleotide pool, then BRCA1 jumps onto these sites of replication arrest in the S-phase nucleus. I didn't understand this phenomenon when I first saw it, but once we had the connection to RAD51 and homologous recombination, once I'd done a little bit of reading, I realized that the connection between homologous recombination and stalled forks and stalled fork repair dates back to the 1960s with seminal work done in E. coli with the REC-A gene. So this was in a sense a rediscovery and highlighted a potential importance of the BRCA gene pathway in HR in stalled fork repair in mammalian cells. This was very much supported by functional data, as shown here, for example, in a metaphase spread of chromosomes from BRCA2 mutant fibroblasts. This was work done by KJ Patel in Ashok Venkataraman's lab in Cambridge, UK. And what we can see in this metaphase spread is that, for example, here we have one sister chromatid that's intact, a second that's broken. So there's an asymmetrical pattern of damage. And that is highly suggestive of replication-associated repair defects. So lots of data all at once kind of conspired to tell us that we need to study the stalled fork, perhaps to understand something about BRCA1 and BRCA2. So we can ask a lot of questions about BRCA1 and BRCA2. Some of them I probably will not have time to address in today's talk, but we can ask at least what functions do BRCA1 and BRCA2 perform at stalled forks, and can we exploit those functions perhaps for therapy of BRCA-linked cancer? It may be that the success of PARP inhibitors reflects a kind of interaction at stalled forks. So I think the community as a whole is very interested in trying to understand the relationships between BRCA genes and other proteins that act at stalled forks. So we now know that the BRCA pathway with RAD51 performs multiple functions at stalled forks, including canonical homologous recombination processes of repair in the Fanconi anemia pathway of replication-coupled repair. But it also performs non-canonical functions. For example, RAD51 is important for fork reversal, for the remodeling of the stalled fork. And the BRCA pathway, together with RAD51, plays an important role in protecting nascent strands from aberrant resection, so-called fork protection, first discovered by Katharina Slacher in Maria Jason's lab. What I'm going to talk about today is a role specific to BRCA1 in preventing the formation of tandem duplications at stalled replication forks. Okay, what is a tandem duplication? This cartoon summarizes how we understand these rearrangements. So what we're looking at on the upper chromosome is an intact structure in which all the letters are appropriate, all our ducks are lined up in a row. And if we were to deep sequence this upper chromosome, we'd find only wild type sequence. In contrast, the lower chromosome has undergone a tandem duplication of elements E and F. So you can see we've got 2 copies of each of those on the new chromosome. If we were to do whole genome sequencing of this chromosome, we would find qualitatively normal sequence all the way along, with the exception of a single breakpoint that brings together a locus downstream of F with a locus upstream of E. And those 2 points where the tandem duplication breakpoint forms defines the span size of the TD, as shown here. Obviously, when we do the whole genome sequencing, we're going to see a change in copy number for E and F. So tandem duplications are in fact the most frequent type of structural variation in the cancer genome. So they're kind of elemental rearrangements, we think, in cancer. Until recently, it was thought that cancer-associated TDs are all of large span size in excess of 200 kb. But with the advent of whole genome sequencing, it's become apparent there's a subclass of cancer-associated TDs that are much smaller than that. This summarizes some data that we obtained in collaboration with Ed Liu and Francesca Menghi at the Jackson Laboratory. I should mention that Ed Liu's lab and Serena Nick-Zainal's lab in Cambridge, UK, co-discovered a phenotype connecting tandem duplication formation to BRCA1 mutant cancer. So what does this data show? What we're looking at here is a series of whole genome sequence studies involving either ovarian or breast cancer, and we've plucked out the triple negative set from this larger Sanger breast cancer sample in the lower panel. Each tick mark along here represents a cancer, and they're arrayed in terms of increasing TD number. So the ones with the largest number of tandem duplications are at the right-hand side, the fewest at the left. On the y-axis, we're looking at the span size of the TD. And you can see this cloud of gray TDs here. The median span size is well in excess of 100 kilobases. In contrast, these cancers have a predominant TD span size of only 10 kb. Okay, this is a log to base 10 scale. So log of 10 is 1, log of 100 is 2, and so on. So these orange ones are full of TDs and they're very small in size. Strikingly, those cancers are almost all BRCA1 mutant, as shown in the red tick marks here. So they're strongly correlated, 95% mutual concordance between loss of BRCA1 and formation of these small, what Ed Liu called group 1 tandem duplications. They're anti-correlated with BRCA2 mutation. So BRCA2 loss does not predispose to these TDs. Okay, the reason I'm telling you all this is that Nick Willis in my lab, who's now a senior scientist in my lab, serendipitously found that he could model tandem duplications in BRCA1 mutant cells. And the way he found into this was by developing a tool for studying stalled fork repair. We're all aware of the incredible power of targeting double-strand breaks to specific loci so as to measure repair products and get an understanding of the genetic framework that regulates repair. We hoped to do the same thing with stalled fork repair. And so what Nick succeeded in doing was generating a tool derived, in fact, from E. coli natural replication fork barrier called TUS-TER, in which a monomer of TUS protein binds very tightly to a simple 23 base pair sequence called the TER sequence. Nick found that if he put 6 TER sequences in a row, then the TUS-TER barrier is a very effective block to forks arriving from either side of the barrier. So the TUS-TER barrier stalls forks in both directions. What's more, by placing the TER array inside of an HR reporter, homologous recombination reporter, he was able to measure homologous recombination and other types of repair at stalled forks. So we need to dig into the method of the reporter a little bit here in order to understand how we're using it to study tandem duplications. So what we're starting with as its backbone is a Maria Jason reporter, okay, in which we've got 2 mutant copies of GFP, one truncated, one interrupted by an ISCE1 recognition sequence and by a 6x TER array, which occupies about 200 base pairs within the GFP copy. We've also got an element here that I'll return to in a moment. And with all of the studies I'm going to show you are done in mouse embryonic stem cells. And in all cases, we've targeted a single copy of the reporter to the ROSA 26 locus of these cells. So I think we can go through this pretty quickly. We're all familiar with the way these reporters work in the case of a double strand break. misaligned recombination generates wild-type GFP. This is the conservative, error-free, if you like, form of HR that dominates in wild-type cells. But as Maria showed some years ago, there are also replicative responses generated in response to a double-strand break, in which the reporter changes its structure. It's now been amplified to include 3 copies of GFP. And the reason this is interesting is that these replicative responses are reminiscent of break-induced replication. And indeed, we think there's a strong, similarity between these processes, especially in view of recent work from Xiaohua Wu implicating the helicase PIF1 in this process. So a BIR-like response is also present in this case. And we wanted to study this pathway, so we developed a tool that allows us to pluck out the long gene conversions away from the more numerous error-free repair pathways. And that's shown here. So that's what this cassette AB is all about. These are 2 artificial exons of RFP, which, as a single copy, don't generate wild-type RFP because the exons are in the wrong orientation. However, in the context of a long-track gene conversion, the AB cassette is duplicated, and now splicing from the first A into the second B can occur, and at the mRNA level, these generate wild-type RFP. So we can identify the long gene conversions by being both GFP plus and RFP plus. In contrast, the short gene conversions are GFP plus and RFP minus. Okay, let's see how this reporter fares when we challenge it not with a double strand break, as we've seen here, but with TUS-TER, with a stalling event. And we're comparing here functionally wild-type cells that have a conditional BRCA1 allele against BRCA1 hypomorphs that have been deleted with CRE, so that they now have an exon 11 deleted hypomorphic allele of BRCA1. And we're also on the y-axis, as it were, comparing a control siRNA with siRNA for BRCA1. So we're depleting either the wild-type BRCA1 protein in this case, or the hypomorphic gene product in this case. And as we work our way down here, we're going from wild-type to the most severe BRCA1 defect. And what I want to draw your attention to is only these products, okay? Not only do we see, a defect in HR, but we also see the emergence of a sort of blush of red cells. So these were quite mysterious when Nick first saw them. So he sorted them, did southern blots, sequenced, and so on. And lo and behold, these are in fact tandem duplications. So the reason we've captured them is very fortuitous, that we have this RFP cassette, which scores positive and turns to RFP plus. But the thing in the middle, the breakpoint, is not wild-type GFP. It's 2 fragments of GFP that have been fused by non-homologous end joining. So this is a tandem duplication. And by a quirk of fate, pretty well at the same time that Nick had the first sequence of this, we made contact with both Ed Liu and Serena Nixinal and found that they were developing their studies on tandem duplications in BRCA1 mutant cancers. So quite fortuitous kind of coincidence, sort of seeing this process both from the cancer genomics perspective and from the basic science avenue. Okay, so in terms of what controls TDs in this system, we found that BRCA1, BARD1, and CTIP, when depleted, boosted TD formation. Interestingly, when we do the same experiment in BRCA1 mutant cells, 2 different proteins came to the fore as suppressors. That's FANCM and Bloom's, 2 sort of storied motor proteins that act at the stalled fork and act on recombination intermediates. So this implied an interesting interaction between BRCA1 and these 2 proteins at stalled forks. This just shows you the data. So we kind of couldn't ignore this, that when you deplete FANCM in a BRCA1 mutant background, you get this rip-roaring induction of tandem duplications. So in terms of specificity, we're able to trigger repair either with TUS-TER to stall the forks or with a double strand break with ISC1, for example. And although we do see a few red responses here, these are not tandem duplications of the type I've shown you here. That's a story for another day. In the stalled fork response, we see that FANCM depletion alone doesn't induce TDs, But here we've reproduced that finding that BRCA1, BARD1 loss boosts TDs, BRCA2, RAVD1 hardly at all. So this seems to reproduce the pattern of specificity that we saw in the cancer genome, that TD suppression is specific to BRCA1. And so we think that the TUS-TER system recapitulates TD formation in BRCA1-linked cancer and might be a useful model for understanding this process. Okay, so let's take a look at FANCM. FANCM is a complicated protein. It has both scaffolding functions and motor protein functions. It's an ATP hydrolysis machine mediated by this motif in the C-terminus. But also, as Andrew Dean showed in Steve West's lab many years ago, plays important scaffolding roles in recruiting the Fanconi core complex to the stalled fork, and also in recruiting the Bloom's top 3A complex implicated in whole-day junction processing and other DNA transactions, the BTR complex, so-called. FANCM has numerous documented abilities to remodel DNA structures, especially branch structures, which I won't be discussing further here. So Arvind Pandey, a talented postdoc in my lab, took a genetic approach. He started by generating what we think of are null alleles of FANCM by engineering early frame shifts in both alleles. And as you see, when he looks at FANCM null cells and depletes BRCA1, we again see a strong induction of TDs. In other words, that's kind of like seeing this genetic interaction from the other side. Instead of a BRCA1 mutant depleted of FANCM, we've now got a FANCM mutant depleted of BRCA1. So that was encouraging. The next thing Arvind did was to try to analyze individual FANCM domains in this function. To do that, he took a novel approach, which Rajula Elango had pioneered, in fact, in my lab, another postdoc who had, with the help of Nick, who developed the Cas9 methods in our lab, made it pretty simple to carve out huge chunks of the genome by dual SG RNA incisions. So what Arvind did was to delete the entire locus of one FANCM allele, leaving just a captive wild-type hemizygote. He then engineered in-frame deletions corresponding to the MM1 domain, the MM2 domain, or the ATP hydrolysis domain, and asked what is the impact of that functionally, studying the impact on the endogenous gene. So I don't have time to show you all the data. In fact, this has been very recently published in Molecular Cell. It's online now. But I'll summarize briefly that Arvind found that if you delete MM1 and thereby prevent the recruitment of the Fanconi core complex and the proper activation of the Fanconi pathway, there's a specific defect in error-free HR, the short track gene conversion pathway. Everything else is unchanged. In contrast, if you delete the MM2 domain, and there Arvind observed a defect in the recruitment of Bloom's to the stalled fork, then those replicative responses of long gene conversion and TDs, in fact, are de-repressed. So these are separation of function alleles that I think have been very helpful for us in helping us to understand the extent to which these different repair pathways can be separated. So I'm going to show you a little bit of data on a mutant that he made that's deleted for key residues in the ATPase domain. So this is an ATP hydrolysis defective mutant. This is a 66 base pair, so 22 amino acid deletion in frame. And as you can see from the Western blot of the endogenous gene products, the abundance of FANCM is not changed when you delete this little segment of the ATPase domain. I'm not going to show you the data, but Arvind also made a D202A point mutant ATPase defective allele, and that absolutely phenocopied the DEAH mutant we're seeing here. Interestingly, the DEAH mutant, the ATP hydrolysis mutant, accumulates at stalled forks to higher levels than the wild-type protein. This is shown by CHIP, where Arvind is using CHIP primers very close to the TER array in order to detect FANCM recruitment in response to a TUS-TER block. So there's an enhanced abundance of the ATP hydrolysis defective mutant. So we think there's something, quite a big clue there to suggest that ATP hydrolysis may play a role not only in DNA transactions at the stalled fork, but also in ensuring the timely displacement of FANCM Put it another way, an ATP hydrolysis mutant form of FANCM may be stuck at the fork and may be interfering with fork functions indirectly through the failure of its proper kinetics. Functionally, the ATP hydrolysis mutant is defective for all the FANCM-dependent repair processes we can measure. So there's a defect in short-track gene conversion, consistent with the Fanconi recruitment defect. As I mentioned, the Bloom's recruitment defect corresponds to an increase in replicative response, and we see the same in the ATP hydrolysis mutant. And in addition, when we deplete BRCA1, we then see a strong induction of tandem duplications in the ATP hydrolysis mutant, less in the wild type. Finally, Arvind attempted to delete BRCA1, that is to delete exon 11, to generate a hypomorphic variant on a FANCM mutant background. And the way he did this was using the Cre recombinase, where he transduces with adeno-Cre and then picked 48 colonies and analyzed the BRCA1 genotype. So this is a sort of busy table, so I'm going to walk through it slowly. Let's start with the wild type cell, so FANCM wild type. When he picks 48 colonies, he retrieves 34 that have not deleted BRCA1 because Cre isn't 100% efficient, and roughly 1 in 3, 14 out of 48, have deleted exon 11. So the hypomorph is, of course, viable on a wild type background. In contrast, when he looks in the FANCM null cells with the 2 frame shift alleles that we defined earlier, he retrieves plenty of clones that did not delete BRCA1, but only 1 that had in fact deleted exon 11. And we verified this by whole genome sequencing. This is not a happy clone. It's growing very slowly. And in fact, if you transduce it with TUS, transfect it with TUS, it generates large numbers of tandem duplications. So this is showing us a genetic, again, from a genetic point of view, that same interaction. Interestingly, those separation of function alleles I talked about, the MM1, MM2 mutants, are viable on a BRCA1 mutant background. In contrast, the ATP hydrolysis defective mutant, the delta DEAH, Arvind was unable to retrieve any BRCA1 mutant clones from that background. So what that's telling us is that FANCM null, and especially FANCM ATP hydrolysis defective mutants, are synthetic lethal with BRCA1 exon 11 mutation. This is in mouse embryonic stem cells. We're at the very early stages of understanding this relationship, but it raises the interesting question of whether this relationship is retained in the cancer genome, and if so, could one generate drugs to target and basically silence the ATPase function of FANCM, and if so, might that be a useful therapeutic in BRCA1 mutant cancer? So our current research is aimed at sort of restudying this interesting synthetic lethal relationship in cancer cells. So to summarize what I've shown you today, we've found that the genomes of BRCA1-linked breast and ovarian cancer contains an amazing number of small 10 kb tandem duplications. That's the seminal work of Serena Nixanel and Ed Liu. We've shown that the TUS-TERS system recapitulates BRCA1-linked TD formation, and that's allowed us to define new interactions between BRCA1 and FANCM and Bloom's at the stalled fork. And by studying FANCM, we think we might have our fingers on an interesting druggable synthetic lethal interaction that exploits this very unique interaction between BRCA1 and FANCM at stalled forks. I put at the end that we have a theory about a possible way that this group 1 TD phenotype that's specific to BRCA1 linked cancer might lead to a bias towards triple negative breast cancer and ovarian cancer that's well documented in BRCA1 linked cancer. But I think, again, that's a story for another day. So I'd like to thank my funding resources, in addition to the Basser Center, who, as I mentioned, had an important role in supporting this research. I've mentioned the people in my lab who did this work. And this is a long-standing collaboration with Ed Liu and Francesca Megni, and we have a number of other collaborations that are not a feature of this talk. So thanks again.